I could not be happier to introduce today's speaker, Dr. Roger Powell, who, when I first met him, was still teaching at NC State.
I am still teaching at NC State.
But still teaching at NC State. So retirement has been in name only for Dr. Powell. Still a prolific scientist, at least 200 publications and dozens of graduate students, so enough to make any researcher jealous. And on top of that, he's a super nice guy. My first encounter with him was when I first tried to get into NC State as a PhD student. And I sort of shuffled around from one faculty member to the next. And when I walked into Roger's office, it seemed to me was almost totally enveloped in this enormous elk rack that was hung on the wall behind him. And to me, it appeared that the antlers were just sort of wrapping around him. And I was speaking to him out of this cage of mammals. And anyway, then later, he and Dick Lansio were co-teaching a grad student seminar. And I knew I was in grad school and had made the right choice when we spent half a semester just kicking around in discussion what a home range was. Like philosophically, what is a home range? And many of my students will know that I've shared your personal home range estimation story, that at one point Roger got so interested in the radio collared himself and estimated his own home range to try to see if he was doing a good job of picking out the important spots. So that's a story though. So anyway, with that, I'll get out of the way and let Dr. Powell talk to us about fisher reintroductions. Thank you, Roger.
Thank you. Now, the first... My first question is logistical. Do you need me to use the microphone, or is it okay if I don't?
Use the microphone, please.
Is that okay? Man, I'm all wired up. Okay, I'm going to talk today about a research project I've had going on fishers in Northern California that started in, well, we actually put fishers on the ground first in 2009. But although it's a presentation about a reintroduction, I hope that when you leave, you think differently about reintroductions than you did when you first came in. Now, I'll be talking about multiple objectives, and I'll be talking about how to do science. Animals and plants have been moved around on this globe for a long time, and in many different ways, and for many different reasons. Oftentimes, they've been reintroduced to places where they have been extirpated to try to reestablish populations. And that's happened in many different ways. Sometimes the number of animals moved is absolutely astounding. In the years of black-footed ferret reintroductions, over 3,000 black-footed ferrets have been released on areas where they used to exist. And over 13,000 wapiti have been moved here in North America. The biggest one I know is over 20,000 sables were moved around the former Soviet Union, trying to reestablish populations and also trying to improve the quality of fur in some of the populations that were still extant. Now, that gave these sable reintroductions two goals. One goal was to reintroduce populations, but another one was to change the quality of fur. So let's think about having dual purposes. For almost all reintroductions, the standard definition of success is if a new population, a self-sustaining population, has been reestablished. That is a single goal, and it's a very confined sort of definition. It's really limiting. It ignores community consequences of reintroducing an animal, as we've seen with the wolves and elk in Yellowstone. It ignores social conflict and human opinions about whether a reintroduction should occur or not, and how humans respond. It ignores ecosystem services, such as fisher predation on porcupines and the effects of porcupines on timberlands. And it completely ignores science and research and information and education. So I think that defining success for a reintroduction simply by saying that you've reestablished a population is very narrow and short-sighted. Reintroductions present the perfect sort of conditions for doing good science. And little by little, people are starting to realize this, as you can see by some of these papers. All translocations have a number of characteristics. First, they put organisms, animals or plants, into novel situations. The situations that are novel to the individuals involved and novel to the release site and novel to populations at the release site. They provide opportunities to control the conditions. The people involved in reintroductions can choose how many animals to reintroduce. They can choose sex ratios. They can choose age ratios. They can choose even specific individuals that have narrow characteristics that they want. Big males versus small males, something like that. They can control the micro and macro conditions at the release sites. They control the habitat at the release site. They can control diseases and parasites. A number of things can be controlled, and these can be used in science. You can compare what happens at the release sites for the population in question, and also what has happened at the source sites where you've pulled animals away. You have foreknowledge of a whole bunch of stuff. You know how much food is available at the release site. You know what competitors are there, what predators are there, and what the habitat is like at the release sites. So everything can be constructed as experiments to test specific hypotheses and learn more about the biology of the animals that you're reintroducing, or plants. And it can be used, the results can be used to inform management actions. So you've got some simple connections. You've got science that can contribute to research success that can then can be used for testing hypotheses, and that feeds back on better science. Under some circumstances, I think that science should actually be the main goal for a translocation or reintroduction. And I'm going to illustrate some of these ideas with research that I've done on a reintroduced fisher population in Northern California. Fishers are members of the Mustelidae, the weasel family, and they are most closely related to wolverines and martens. There's a large sexual dimorphism in body size, where adult males weigh 2 times or more as much as females. There you get to see their faces compared to a human. Fishers are predators, and they prey on diverse prey, but the most important prey for them are, well, the most important food is probably road-killed and hunter-killed deer. But beyond that, starved deer in the wintertime, porcupines, snowshoe hares, and then some smaller mammals and diverse birds. Porcupines are a particularly interesting prey for fishers. Fishers are the only predator that is highly evolved to prey on porcupines. They have highly evolved specific behaviors, and wherever fishers and porcupines coexist, porcupines are a major prey for fishers. They kill them by circling a porcupine that they can get into an open space, and then they kill it by attacking the face, which is the only part of the exposed porcupine's body that isn't protected with quills. It's a lot of work. It takes a long time for a fisher to kill a porcupine, but if the fish is successful, it's got over a week's worth of food. It's a really important food source. Fishers are generally found, they have their highest population densities across North America in old-growth forests. Forests that have never been logged and show little effect of humans. Female fishers den in spring around right about now, and they exclusively produce kits, produce their litters in tree cavities, a cavity in a live tree or a snag. They never produce kits anywhere else. They're females that are 2 years old or older give birth around now, and then they breed within 10 days. And they have delayed implantation, and then they implant in mid-February and produce kits the following April. The cavities that females use are just the right size for a female fisher to get through. They almost exactly match the head size of a female fisher. And here we have a female sort of blocked by some vegetation, and you can see her two kits, which at this time have reached about 12 weeks old. Fishers have a long history of interacting with humans. The Anishinaabe knew that it was the fisher that released the birds that were held by the evil sky member, and in order for the fisher to escape from being killed by that fellow, the fisher had to jump into the sky. This hoopoe boy is holding a quiver that's used in ceremonies. Quiver is made from a fisher pelt. And interactions with humans, with Europeans, has been going on since Europeans arrived. You can see that the original range of the fisher is shown by this tan stippled area, and interactions with humans, trapping and logging, led to a reduced range outlined in blue, and you can see the present range in pink. Fishers have gorgeous pelts, and they've had some of the highest pelt prices in history. And they depend on old growth forests, or at least old second growth forests, and the changes in forests, that extensive clear cutting in the early 1900s destroyed a vast extent of fisher habitat. Fisher reintroductions began in the 1930s and were actually fairly common from the 50s to the 80s. Fishers have been reintroduced over 30 times, and the two most recent reintroductions, well, the three most recent, two have been in Washington, in Olympic and Cascade National Parks, and then the reintroduction that I've been dealing with in the northern Sierras of Northern California. They've been introduced specifically to control porcupine populations. That happened mostly in the 50s and 60s. Porcupines can change forest composition by over-pruning trees, especially hemlocks and white pines, and also by debarking trees, especially sugar maples, red maples, and basswoods in the spring. Only one fisher population has been monitored along with a porcupine population to see if fishers actually cause the desired effect of reducing porcupine populations. And you can see in this one site in Upper Peninsula Michigan, fishers were reintroduced in the mid-60s, and the porcupine populations went down over the ensuing 25 years. Now, that would imply that the fishers may have had an effect on the porcupine population. Unfortunately, this result is confounded with a change in the availability of winter den sites for porcupines. So fishers arrived, and winter den sites declined in occurrence, and so porcupines were actually hit with two things at once, and we don't know how big the effect of each of those two was. So fishers did kill porcupines. I know that. I saw where they killed them. So only one site have both predator and prey been followed, and in that site we still don't have a real good understanding of everything that went on. Now, in the early 2000s, Sierra Pacific Industries, a large timber company in California, suggested that fishers might be reintroduced on some of their properties in the northern Sierras. And they went to California Fish and Game Department and the U.S. Fish and Wildlife Service and proposed that fishers be involved in this reintroduction. And luckily, they got in touch with me. Luckily for me, they got in touch with me and involved me in planning the reintroduction and in doing research once the reintroduction started. Between 2009 and December of 2011, we moved 40 fishers from the range of fishers in northwestern California to our site, our study site, which is in the northern Sierras. It's right where the northern Sierras meet the southern Cascades. It's the red oval on this slide. The reintroduction has from the start had multiple objectives. One is to monitor fishers on a managed landscape where timber production is an important aspect. So we weren't trying to manage the landscape for fishers. We were trying to learn how fishers would deal with a landscape managed for timber production. We wanted to evaluate the primary reasons for success or failure to establish a new population. We wanted to test models of habitat suitability and have a series of hypotheses related to the types of habitat models that have been developed and also the specifics of those models that are related to resource requirements of fishers. We want to test models on optimal home range choice by fishers. And we wanted to test the effects on a source population of pulling a number of prime individuals, those that are at the peak of their reproduction, out of those source populations so that we could take them someplace else. Does that really have an effect on the source population or not? We outfitted every fisher that we moved with radio collars. Most of them were VHF collars that just gave us beeps, and we had to get out on the ground to locate the fishers. But we put Argos collars on many of the adult males, and we used some GPS collars at various times for both males and females. We put remote cameras, especially marked on fisher dens, to document where fishers went, and for those on the dens, how many kits each female fisher had. So you can see here's a female moving one of her kits that's about 10 weeks old, and you can see the camera in the background. This tree, this den actually had 3 cameras, and the photograph that shows the back camera was taken by the front camera. And here's a female, another female who's moving a kit. We know that this female had at least 3 kits because a photo taken after this one shows the female carrying 2 kits in her mouth at once. So our counts of litter size are minimum counts. We know at least how many kits we saw on the camera, on the photos, but she might have had some that she fooled us and moved without us seeing it. We got to document other carnivores using the cameras, and spotted skunks, gray foxes, and ringtails are the 3 other similar-sized carnivores that are found in our study site, similar sized to fishers. And we could document prey. Gray squirrels turned out to be the most common prey for the fishers on our study site and make up 60 to 70% of the biomass that the fishers are eating. So here's a female fisher taking a western gray squirrel up to her kits. So she'll carry it up the tree. And I have to say thank you to all the folks that have helped me on this project. I couldn't have done it without all of these people. But the ones that are here are ones that have been graduate students or full-time technicians, lead technicians on the project. So now let's look at doing science with this fisher reintroduction. The first thing I want to talk about is population trends for this reintroduced population and trying to understand why the population in its size did what it did. Well, here's the basic population trend. This is the minimum number of females that we have documented each year, starting in 2009 up to last fall. And you can see we went from the first 9 females we released in 2009 up until we had in the upper 60s of known females on the site in last October and November. Now let's get a better idea of what's going on. By doing population modeling, we've been able to determine that the 2 critical variables are litter size and juvenile mortality. You can see that those 2 have the biggest effects on the on population growth, and that mortality, actually this is the flip side, it should say survival. When survival goes down, that has a huge, juvenile survival, that should have a huge effect on the population. Now, we have documented deaths and we've documented reproduction. We know that the denning rate is 82% of females establish dens each year, and that the minimum average litter size over the whole period is 1.8. We know that the survival to the first autumn is about 0.9. And we also know that survival of kits is heavily dependent on survival of their mothers. And adult females have their highest mortality rates, their lowest survival, during the period when they have kits. And that mortality is due to a number of things. And this This photo shows one of them, and you can just barely make out there's sort of a blob right here, which is a female's head, and she is in an old water tank. Now, this area, our study site, was logged in the late 1800s and early 1900s, and most of the roads that are, main roads that are out there now were railroads back then. They were narrow gauge railroads, and there were water tanks and water barrels set out across the whole area. This is one of those old ones that still holds water. And a female fisher who had at least 2 kits in her den, got into that thing, we don't know why, but she couldn't get out and she drowned. So we get different sources of mortality, some of which were very unexpected to us. We have found that the major sources of mortality are actually bobcats, roadkills, disease, and rodenticides. Northern California is a major site of illegal marijuana grows. And the marijuana growers just inundate their sites with anticoagulant rodenticides and other rodenticides that they can buy without needing permits at any Lowe's or Home Depot or any of the other big warehouse stores. They can back a truck up, fill it with rodenticides, take it off into the woods, and fishers are dying from the poisons that are killing the rodents that come to those sites. So if we look at what the fisher population has actually done, which is the short brown line, you can see that the fishers appear to be doing a little bit better than our predictions for the fisher population based on, one, what are the reproductive rates and mortality rates that we've documented, and two, for the first year mortality rates for the juveniles. And we don't have any good direct measures for juvenile mortality during the first whole year. And the estimate we have of 0.6 survival, or 60% survival, 40% mortality, has very, very wide confidence intervals. So we have run models with 30%, 40%, and 60% survival on here. And you'll note that we can't differentiate whether a population will roughly stabilize or grow until we've got about 10 years worth of data, and we don't have quite that many years. We've got 8 years. So the models suggest that we need at least 2 more years of data to be able to say securely that the prediction is population growth. The actual population growth that we have documented is a little better than our predictions. And what this suggests is that we have actually established a fisher population on the site that is independent of the original animals we reintroduced. It is self-sustaining under the present conditions. But we haven't done the analyses yet to be able to look forward and say, with the logging plans that Sierra Pacific industry has, whether that fisher population is predicted to continue to grow or not. So I'm confident to say we established a population. It's there. If things don't change, it has a good chance of staying there. We still have possible demographic and environmental stochasticity that could drive it extinct or could really help it. Second example of science is looking at the probability, how to increase the probabilities of reintroducing a fisher population, and then extending that generally to how can we maximize the probability of reestablishing any population. But going into this reintroduction, before we let any fishers go, we hypothesized that fishers, that reintroduced females need to establish home ranges. They need to explore their environment, find where the food is, find where the prey are, and find potential cavities for giving birth. And they need to do that before reproduction. And that if we let them loose too close to the time of implantation, that lots of them would not implant and would not produce litters. So the original plan for the research was to release every year all of the fishers designed for that year's release in October, November, and December. And that all before the first of the year, giving them three months at least to set up home ranges and find cavities. So we collected data on our fishers, and we found that even though we wanted to release them all before the 1st of January, reality crept in, and sometimes we couldn't. And so in the first 2 years, we released some of our fishers in January, and we even released 3 in the first 2 days of February. As I said, that hadn't been our plan, but it did set up an experiment. We had an a priori hypothesis that we shouldn't let them go after the 1st of January, and son of a gun, we could now test it. And then we were able to share data, get data from the reintroduction of fishers to the Olympic National Park. And so we could use 2 study sites, 2 reintroductions to test this hypothesis. So the distribution of releases, you can see, for my study in California, most of them were done before the 1st of January, but some after. And then for the Olympic releases, most of them were actually done after the 1st of January, and some of them were even released in March, almost to the time of giving birth. If we look at the timing of release and the proportion of females denned, you can see at both study sites, more females released early denned than females that were released late, and that there is a big study side effect. So in Washington, more released early had kits than released late, but in total, because so many were released late, and we kept our fishers in captivity only as long as it took to get a disease profile on them and basically a disease clearance, a health clearance. So most of our fishers were released 2 to 4 days after we caught them. For the Washington reintroduction, most of them were held for 2 to 4 weeks. So there was a big difference in handling there too. You can see there's a 59% reduction in reproduction for our fishers, and a 73% reduction for an average of 66%, 2/3. That's a massive effect on reproduction. Fishers that were released after the 1st of January had only 1/3 the chance of producing kits that first year. And you can see that it has an effect on projected population growth. And fishers with early release and late release had different projections for population growth. And if the reduction is 66%, which is what we saw, you can see that there's an effect on population size that goes on into the future. We modeled even a 90% reduction, but you can see in all of them, releasing fishers without giving them time to reestablish home ranges has the potential to cause long-lasting effects on population growth. So, a key here is to understand well the biology of the animals or plants that you're reintroducing, and to understand some what might be unanticipated effects on population growth. Now, if you look at the history of fisher reintroductions, I told you there were over 30 reintroductions have occurred. Most of the ones in the eastern half of the US have been successful, like 80%. Only 40% of the releases in the West have been successful. And there's actually been a difference in the way that fishers have been handled in the releases in the two places. And in the East, they've tended to be released relatively early in the winter, and they have a short handling time. And in the West, many of them have been released later in the winter and have had longer handling times. Now, those data are much sloppier than the data for the two sites where we do have good data. So I wouldn't want to say that this effect has affected all of the releases, but I think it's something that's been ignored in almost all the releases. And to be honest, I am flabbergasted by what's going on in the Cascades right now, where fishers are being released in Cascades National Park, and most of them are being released in February and March. They're completely ignoring this result. And not only that, the guy who's leading the project is a co-author on this paper. I don't understand what's going on up there. Okay, a third example, and this is spacing patterns of fishers, and how competition between conspecifics and how resource depression can make a difference on where and when fishers establish home ranges. Now, you may not be familiar with the term resource depression, and I'll define that in just a little bit. But certainly we know that competition is important, both to animals and plants, but certainly for carnivores. And oftentimes in very graphic ways it is important. And it's important between conspecifics. Just about any good movie that shows about wolves does show members of the same pack even scrapping over food and having arguments over food. Now, resource depression is a term that's used to describe changes in prey availability even when prey aren't killed. So you've got resource depletion, which comes from a consumer eating something. But resource depression comes when a predator makes prey alert and vigilant, and the prey look out for predators and stay away from places where predators might be. So that reduces their availability, even though their population size doesn't go down. Well, an individual critter benefits from maintaining a home range. The resident of that home range knows where the resources are, those really important resources like couches. Intruders risk experiencing resource depletion and depression. So when a resident of a home range goes into a place where there are lots of prey and searches, and maybe it catches something and maybe it doesn't, it's having been in that site with dense prey has alerted those prey, and they're on guard. Another critter that does not have the same home range, but that intrudes, doesn't have the history of where the prey have been alerted and where they haven't. And so foraging success is going to be low for that intruder, not just because it doesn't know where the prey are, but when it finds them, it doesn't know if this is a bad place to be because they're alert. So the owner of a home range has a lot of reasons, a lot of benefits for maintaining that home range. Now, what is a home range? It's the area where an animal lives and where it maintains its familiarity with resources. Now, I don't want to go into the weeks of discussion that Jason and I had years ago, but in my head, a home range is something that is inside the head of a critter, something that it knows. And it's not a line we put on a map. And it's not an analysis of probabilities of where the animal's going to go. Those are our analyses, but they're not the home range. I won't dwell on this, but I just wanted to say it. A territory is an area where an animal has exclusive use or priority use, and so it is a subset of home ranges. All critters basically maintain home ranges, but not all of them maintain territories. Fishers maintain territories, and they advertise where they go mostly by scent. They have scent glands on their paws, and wherever they go, they leave scent. When they get up on a log, they love to get up and run along the top of a log. And when they do that, I'm certain they can smell stuff from further away, they can hear things from further away, and they're putting their scent up on a high spot. A fisher encountering an established territory of another fisher should react quickly and leave that territory, because this is not a good place to be. It's going to be hard to find prey. And it might get stomped on by the resident. But suffice it to say, we don't have to have active defense here. All we have to have is insecurity about food for an intruding fisher to want to get out of the territory. And there are differences between the sexes related to territories. We hypothesize that females view territories of both males and females, all other fishers, as having depressed resources and the possibility of being defended by the owner. Males may force females to tolerate overlapping territories to gain mating opportunities. Males weigh twice as much as females. Remember that? They're big. And if a male wants to live someplace, there's nothing the female can do about it. So we hypothesized that males force the females to tolerate territory overlap, to have intersexual territories. And we hypothesized that both sexes should avoid the home ranges of fishers that are larger, older, and dominant. So males that are bigger than females but aren't fully grown yet should also be wary of adult males. Specific hypotheses are that fishers released into an area occupied by no conspecific should travel a short distance and establish its home range in as good a place as it can find close by. Female fishers should avoid the territories of all other fishers, but males might impose or should impose territory overlap onto females. Well, we had the perfect setup. We had a site with no fishers. We had looked at that site and we'd evaluated the habitat that was there and predicted that this should be decent habitat for fishers. And then we let fishers go onto it. And the first year, we let the fishers go where you see the red dots. Oh, the background map here is not just random colors. Yellow is bad habitat, and blue is good habitat. And this is developed from a good model that we've tested. In year 2, we released fishers at the blue dots. And you can see that some of those blue dots are really close to the red dots and some aren't. And then the last year, with the white dots, you can see that, again, some of them are close to where we'd released fishers the year before and some are far away. We were testing this hypothesis. We were purposefully releasing fishers right smack in the middle of established home ranges and then releasing other fishers way far away from established home ranges. So we released a total of 24 females, 16 males. We released about half of each inside the home ranges of other fishers. And then after fishers were released, some of them wandered into the home range of another. So 75% of the females and about 60% of the males ended up encountering, in some way or other, the home ranges of others. And we call a colonist a fisher that's established its territory, and an intruder a fisher that goes into an established territory. And you can see for year 2, some of those blue dots are inside the established territories of fishers released the previous year. And here is a mind-boggling view of dots, but red, blue, and yellow relate to first, second, and third year. And you can see that the red dots really cluster towards the middle, especially for the females. The blue dots are kind of closer, and the dots that are furthest out are the locations of females that we released the last year. So this is evidence that females especially, but also males, females especially released late went further. Now here are a couple examples. 3 females in year 2, and you can see that the blue female was released in the yellow territory of a female from the previous year. That blue female scooted right away. The same sort of thing happened with the black female. After we released her, she walked right into the green territory, and then she scooted through a whole bunch of others and finally settled down away from any previous territories. And the pink gal down on the bottom, we released away from a territory, and she basically settled where we let her go. So these are examples. Males do a different thing altogether. What they did was highly variable and depended a lot on whether it was a big male. Almost all big males tended to settle down unless they were settling down where there was no female. And then they went searching for females. Young males that we let go, even though they were way bigger than females, tended to avoid where females were. They would settle down right where they were, where we let them go, unless a big guy came along. And then some of them went long distances. We had one juvenile male wearing an Argos collar. So this is a collar that tracks itself, talking to satellites every day. We let this guy go for a month and a half. He just stayed right where he was. It seemed like a great place for him to be, at least he thought. And then we let a big male go right there. And this big male was a big male compared to this young guy. The young guy picked up and went down into Central Valley. He spent 2 weeks in the almond groves. Then he came back up. It was really a good thing we had a satellite collar on him. Otherwise, we never would have been able to follow him. Okay, so here we have some quantified data from all of the animals that we released. And you've got blue lines, which are colonists. These are males or females released where no fishers have established territories. And we have intruders. Those are the black lines. And they're released into sites that had established territories. And we have a whole bunch of different ways of measuring how the fishers moved when they were released. One was we started with the release site and then built convex polygons that fit all of the points where the fisher went. And so a fisher that explored a huge area before settling down would have a big polygon, and one that didn't explore a huge area would have a small polygon. To the upper right, you've got distance traveled. This is the mean distance that it traveled from the release time to any given location. And what you see in both of these for females is that female colonists have smaller measurements, smaller areas explored, and smaller mean distances moved than female intruders. And for males, there's a more complicated story that, as I said, has to do both with where the females are and whether the males are big or little. Another is distance to the home range center, where the fish are finally settled. You can see again that the colonists have a smaller measure. They settle down closer to where they were released. And finally, the maximum distance traveled again. The whole story is the same thing in all of these different measures. Females that were released into an area with no other fishers settled down right where they were, very close. And we have data on habitat quality, and the mean habitat quality of where we released them and where they settled is basically the same. So they weren't choosing on habitat quality. A major thing was whether there was a competitor or not. So the conclusion is that female fishers avoid the territories of all other fishers, as we hypothesized. Dispersing young females, I didn't show data for those, but they did exactly the same thing. When they took off to disperse, they'd go through established territories till they found a space that was open. And if a female close by died, they would go to that spot and settle down. So the same pattern that we see for the released fishers. Male fishers imposed their territories on top of females. So they did not avoid other fishers. They tended to avoid other big males. But that was the only thing they avoided. And so as a consideration for reintroductions, you need to understand where you put animals, how long they stayed there, where did they establish themselves, and you need to understand this related to the sexes and the different strategies of the two sexes. And finally, you have to realize that augmentations, where you add animals to a site that already has a population, but for some reason you want to increase that population size. Yeah, we won't get into my reservations about augmentations. Anyway, you have to realize those are different than reintroductions because of the residents that are there. And they have an effect on what the released animals will do. So I could go on. I mean, I'm running out of time, but I have further examples of hypotheses we tested related to habitat, related to energetics of the fishers, related to the source population, and then more management implications that all stem, a lot of which insights we would not have gained if all we had done was let fishers go on this landscape and then just followed now and then to see if they were still there and maintained a population. We've learned a lot about basic fisher biology, about why fishers in all populations have the spacing patterns they do, and why probably most others, solitary carnivores that have the same kind of spacing patterns, I think our data apply to those other carnivores as well. And we've increased our understanding of the importance of habitat and movements on when fishers will have successful reproduction or not. And doing science well beyond just releasing fishers has really helped us understand fishers and other carnivores better. So with that, I'm happy to take questions.
The relationship between fishers and coyotes. The coyotes prey on fishers, and I've also read that where you find fishers, you won't find feral house cats. They kill the cats. Is that that?
Okay, there are two questions here. One related to fishers and coyotes and predation, and the other is fishers and house cats and predation. Coyotes do kill fishers, and we have coyotes in our study site. Nonetheless, we've only documented one coyote killing a fisher, and we've documented 6 to 8 bobcats. There's for some of those we can't distinguish between bobcat and mountain lion, but we know it wasn't a coyote. So coyotes will kill fishers, but in most places they're not as important as the felids. The only site, fishers have recently colonized on their own the cities of Albany and Schenectady in New York. They're found inside the city limits. This is an unexpected occurrence, but it's really cool. And a guy named Scott LaPointe has studied those fishers, and he collected scats and looked at diet. Nowhere, anywhere, have house cats cropped up in fisher scats, and specifically inside those city limits, the fishers did not kill cats. This is really a disappointment to me. Actually, I had a reporter call me once with exactly that same question. Had to do with fishers starting to colonize Connecticut, and did people need to worry? And I said, no, I really wish they did. And she was speechless. She could not, that's not what she expected me to say. And that led the conversation to a completely different area than she'd anticipated.
I read that once where they just kill all the feral cats.
No,
I'm hoping that was true.
No, it's not true. I wish it were. Okay, question.
Earlier in your presentation, you had that juvenile mortality or juvenile survival was pretty much your key factor for keeping an established population. Is there any knowledge between that and the degradation that would happen if your company decided to start logging that area with your established fishers? Anything that might influence the habitat versus juvenile survival?
Yes. As I said, our major source of, it's not a majority, but the highest, the most common mortality factor is bobcats. Bobcats travel roads and use roads way more than fishers do, and clear cutting sites put in roads, and the clear cuts make better bobcat habitat, and they destroy fisher habitat. So bobcat predation is encouraged by the clear cutting that's done. And with the history of, I haven't gone into the complex history of this site, but for the next 40 years, the frequency of clear cuts on the landscape is going to increase. Finally, it'll stabilize, but it won't do that for about 40 years. And really interesting analyses are going to be coming over the next 2 years once we dive into projecting the cutting that's going to happen on the site and looking at available habitat for fishers and then how many fishers that can support. That's going to be some interesting analyses and modeling. Yes?
Your, Dr. Powell, your release site within the original home range, and what is the interest in your paper company for fisher reintroduction?
Okay, the first question is our release site within the original range of fishers? And yes, it is. There's some people that question how dense the fisher population was back before the 49ers came. So we have to go back that far to look at the original landscape. And knowing what the forest was like then, the density of fishers at our site then was probably moderate, not great, because it was a different kind of forest. It tended to be a fire-dominated forest with a relatively open canopy and a lot of shrub layer in the understory. So that would be okay for fishers, but not great. Your second question was why the hell did they want to have fishers on their land? Okay. In 2004, the fisher was proposed for listing under the Endangered Species Act. And the Fish and Wildlife Service determined that that listing was warranted but precluded, meaning fishers deserved to be listed as threatened or endangered, but the Fish and Wildlife Service did not have the resources to determine habitat requirements, limiting habitat, to develop a plan, and therefore fishers could not be listed because they couldn't list them without these other things. They were added to a list of some 400 species or populations. And the Fish and Wildlife Service had not been funded adequately to be able to do the work needed to get the critters onto either the endangered or threatened list. When that happened, Sierra Pacific Industries had fishers on some of their land, and they knew that if fishers were listed, it would affect their management of those lands. They proposed putting fishers on sites that didn't have fishers because they wanted to develop an agreement with the Fish and Wildlife Service so that they would have experimental populations on their land and therefore wouldn't be affected if fishers were actually listed. And their initial step was to say, we want to list the fishers on all of our properties as experimental populations. The Fish and Wildlife Service didn't go along with that, but at this point, Sierra Pacific Industries has actually committed themselves to doing a fisher release, and they wanted to learn what we could tell them about their proposed management for their land, and so they went forward with it. It's sort of a complex answer, but it had to do with politics. Anything else? Yes?
Are there any fishers in Portage County?
Are there any fishers in Portage County is the question, and the answer is yes, absolutely. Their populations over the past 20 years have been extending further and further south, and Portage County is not the best fisher habitat, but it's habitat that does allow fishers to maintain a population. Nobody knows how much it is fed by source populations to the north. It does have porcupines here, and so fishers here are killing porcupines, and that suggests that the population, although it may be low, could very well be self-sustaining.
Do fishers prey on rough grouse?
Do fishers prey on rough grouse? And the answer is absolutely. Whether they affect the grouse population, we don't know, but they certainly do. They certainly do kill grouse and enjoy them.
Will they prey on turkeys?
I don't know about turkeys. My bet is if fishers prey on turkeys, it's only going to be big males. That would be my guess. I think turkeys are too big. Well, okay, one last.
Do fishers hunt in the canopy like a pine marten, or they ambush things on the ground? How do they operate?
Do fishers hunt in the canopy like pine martens? Fishers do not hunt regularly in the canopy, and actually neither do martens. Martens and fishers do both kill red squirrels, and most of the times they catch them in their middens on the ground. Fishers climb trees readily, and I've got a really cool photograph that came from one of our remote cameras that our head technician had aimed up at a natal den that was about 10 meters above the ground. And we've got a picture of the female leaping from the den entry to a nearby tree. And she is stretched out and absolutely in midair. It's just the coolest photo. So they are really agile in the treetops, but they're big. So they aren't able to catch much in the canopy. I've seen some photos of fishers chasing squirrels mostly up and down trunks, but not up in the trees. Most of their prey they catch on the ground. Thank you very much. If you got more questions, feel free to come down.